řeč bude o tom, že na Jižní Moravě máme glaciální refugium. Tak určitě dorazte, protože to je velká novinka, přednáška bude zajímavá. And now I'll switch to English, and it's my great pleasure to hear Professor Pfaus, who did his PhD in British Columbia, if I'm not mistaken, and then for a long time he worked in Canada in University of Concordia, which is Montreal, and he's well known for his research on various aspects of sexual motivation and neurophysiology of sex, and he also held many posts in academic life, but very recently he moved to the Czech Republic because, to put it simply, he had enough of North America,
and I got lucky.
And with all his knowledge, he now is based at the Charles University and Faculty of Humanities, but also in the National Institute of Mental Health. And today's talk will be about how our previous sexual experiences shaping our motivation and pleasure. And the talk is called Cupid's epigenetics. So,
thank you. Well, thank you all for coming and for bearing with me in English because my Czech is awful. I know a lot of adjectives at this point, but not a lot of sentence structure. Anyway, that said, okay, so let's get into it. Why am I talking about Cupid's epigenetics? Clearly, Cupid had no idea about epigenetics. But if you know who Cupid was, Cupido is Latin for passionate desire. Okay, and of course, the Greek version of that was eros. And here's Cupid as a kid, where we usually see Cupid in this form, but this was Cupid and his main love, who was Psyche. So if you think about it, passionate desire and the soul come together. And why do they come together? I mean, if you look at Jacques-Louis David's painting, this is clearly beautifully post-orgasmic sex. Okay? And the son of Venus and Mars, who was Cupid, was Psyche's first love. His child with Psyche was named voluptas, which is sensual pleasure. So you have passionate desire plus the soul equals gives rise to sensual pleasure. But it's also the case, and here's another version of them, but she's always in this like post-orgasmic beautiful pose. It's also the case that pleasure in turn reinforces desire. Okay? And that was clear in both Roman and Greek mythology. So that kind of gives rise to this idea of learning. If you're talking about reinforcement, then you're talking about learning. So what's the relationship of pleasure and desire, which the Greeks and the Romans and many other cultures just simply took for granted? So let me ask it another way. What do we or other animals really need to learn about sex? Aren't our preferences and desires and behaviors innate? Isn't that what we're taught? Don't we just all do it, right? With kind of innate, if you will, variable action patterns that tell us what to do, and I think you can clearly see from the photos that we have here, the answer is yes and no. Some things are innate, some things are not. Pavlov comes to mind because he said in his collected works or selected works, if you want new ideas, you should read old books. Why did he say that? Because everything that goes around comes around, and when it comes around, people reinvent the wheel. So what are some of the reinventions of the wheel here? Prior to Pavlov, Richard von Krafft-Ebing wrote this book, Psychopathia Sexualis. It's all about fetishes and paraphilias around the turn of the last century. And he wrote, of course, in 1886. And predating Pavlov, von Ebing wrote that erotic fetishism makes an idol of physical or mental qualities of a person, or even merely of objects, because they awaken mighty associations with sexual pleasure. And even before him, there was Stendhal, who was this guy, Marie-Henri Beyle, who in France, who said, as part of his principle in De l'Amour from 1822, if the couple remain in close facial contact after the generative act, which of course means sex, he, She was another issue. But he shall come to associate the flower of desire with her features, that to him shall be salient, and he shall seek out similar features in another should this bond be broken. So what Bale is saying is that he has a type. He has a physical type. And if you think about it, to go from distal to proximal to interactive in a visually acute individual, you got to be able to see something that looks hot and then move toward that using some kind of proper social skills. Bale also said that beauty is only the promise of happiness. Again, this notion of associative strength that yields an expectation of some event. He also called the process by which this happens, which is a word that I think we need to reintroduce into neuroscience, crystallization. What he calls crystallization is the operation of the mind or brain that draws from all that presents itself the discovery that the loved object has some new perfections. Something is different. Something is better. What's better? What's different? How does that all work? Now, one truism when we study sexual behavior is that animals and humans seem to get studied very differently. And part of the reason for this is that there are really two types of sexual systems. There's a reflexive system, which involves looking at autonomic activation, genital blood flow, genital reflexes and sensations, innate copulatory behaviors. And these really involve, in the brain, the hypothalamus, the brainstem, and the spinal cord. You don't even necessarily need a limbic system for this. These are what we typically study in animal models because it's those structures that steroid hormones bind to that essentially sensitize the behavior. So when we look at something like mounting behavior in a male rat and lordosis in a female, these are hormonally driven behaviors. We think the animal doesn't think about this, the animal just does it. So all the rest of the brain, the limbic structures, cortical structures, even fine motor coordination, which clearly this involves, are not taken into consideration. Very few rat people study the cortex. On the other hand, the incentive system involves Pavlovian associations between sexual stimuli and reward or punishment. So you can imagine the stimuli become associated with the unconditional stimulus, which is the reward or the punishment, and its representation in the brain. And operant associations, response reinforcer associations, between what you do sexually and how good it is or how bad it is, the reward and punishment that you get from that. And those, of course, involve limbic and cortical structures that interact with certain hypothalamic nuclei, but those are what are typically studied in humans using things like functional magnetic resonance imaging. When you're looking at the brain, looking at a response to an erotic visual cue, erotic audio cues. We look there because fMRI, even to this date, and PET scanning as well, can't really, I mean, it can look at the hypothalamus and say, oh, the hypothalamus is activated, but it can't tell you what region of the hypothalamus is activated. You can't get molecular with it and look at the activation of second messenger or genomic things in neurons, in humans, at least not yet. So, well, okay, so we started out asking the question, what's rewarding to a rat? And how do we know? Do they even like to have sex or do they just do it? Well, it turns out that when we use what's called a conditioned place preference paradigm. Okay, so rats typically like to be in the dark, so they love to be in here, but if you bait the light side with something good, it could be heroin, it could be the period after an ejaculation in male rats, whatever, finally you put them into the middle and say, which side do you spend more time on? If they've made an association of this side with something good, then they shift the preference from here to here. Okay, and if not, if it's something aversive, they'll just stay on this side. Right? So we know that conditioned place and partner preferences are induced by the pleasurable state after ejaculation in male rats. We know that they're induced in female rats by paced copulation. That's copulation that she controls the initiation and rate of. And there are certain chambers, and I'll show you those, that allow that to happen. We can also give her manual clitoral stimulation. I'll call it CLS for now. That's distributed in time in a way that mimics the preferred rate that a particular female likes. How do we do that? Easy. A number 4 fine camel hair paintbrush. That's applied the same way the male's pelvis applies clitoral stimulation when he mounts with intromission, right? Moreover, both females and males emit calls in the kilohertz range that we can't hear that signify an appetitive state, so a high call in the 50 to 60 kilohertz range that forms trills and flats, and a low call within the 23 to 25 kilohertz range after ejaculation or after an orgasm-like response in female rats. Yes, female rats have orgasms, so do male rats. So they show this. So what do we know about males? We know that sexually conditioned place preference, CPP, and conditioned ejaculatory preference, CEP, are formed that the state induced by ejaculation is necessary for the conditioning. So the male rats have to ejaculate and actually be, for the case of CEP, I'll show you this in a second, be with the female. So Stendhal was right, even in rats. So you get a stronger place preference and partner preference when the males are more aroused prior to ejaculation. The acquisition of both of these is blocked by the opioid receptor antagonist naloxone, but it's not blocked by dopamine antagonists. So here's a case where we're looking at looking at place preference and then at partner preference, where we have, when we use an odor, because rats live in a largely olfactory world, so we use an odor that's a conditional stimulus to create the partner preference. Here's how we do that. Odor conditioning of sexual behavior and mate choice. So we'll have, for example, receptive paired males where we smear the female back of the neck and her anogenital area with almond odor. It could be lemon odor, that works. Wintergreen works, as long as the odor is neutral. Okay? Odors like cadaverine, which are really disgusting, or for some reason vanilla, which they love, are not ones that you can use because they're not neutral. They either love it or they hate it. So we use a neutral odor that's paired with receptivity, and then no odor when we give them a non-receptive female. And they get sequential pairings of this. So they learn that when she has an odor, that means yes, and when she doesn't have an odor, that means no. The non-receptive paired trained males learn the opposite association. When she has an odor, it means no, and when she has no odor, it means yes. Okay? And then random paired, you always have to have a random paired group in Pavlovian conditioning, then get the odor sometimes meaning yes and sometimes meaning no. Okay? Ultimately then, we give them kind of a triad copulation test, a little menage a trois, where the male gets to pick between two receptive females, one that's scented and one that's unscented. Now, if we think that the male has, in this group, has been with the scented female, and he wants to spread his genes far and wide in the pool, if he's the shark in the pool, who's he going to want? The one he's had before, or a different one? Well, the Coolidge effect would suggest he's going to then want the different one. Yeah? And likewise for this male, he's been with the unscented, so he's going to go after the scented because she's different. Yeah? What do they do? We train them in what's called a bi-level pacing chamber. So this is a chamber that you can run from level to level. The female's got the odor in the back of the neck and her anogenital area. Those are two places the male puts his nose. Here's the female, there's the male. Here she is making a solicitation. So she hops over him, lets him sniff and lick her anogenital area. Then he kicks him in the face and runs away. It's kind of like grade school. So here she is running away, and here he is following her. And finally she holds a lordosis crouch and allows him to mount with penetration. Typically he dismounts, she runs up, then she solicits by running back down and running away. And so they do this many times before he finally ejaculates. When we test them, we put him in an open field. And here's the pair-trained male, and he has access to two females, one scented and one unscented. The male's under my former grad student Todd Kippin's arm. And here you can see the male going after the scented female. So, what do we find? We look at who he picks of those two females for first mount, first intermission, first ejaculation, and then we look at the distribution across the final 30-minute test here of mounts, intermissions, and ejaculations. Okay, what do we find? Who he mounts first, intermits first, doesn't seem to matter. But look at this. He's ejaculating preferentially with the familiar female. Okay? Here's the pair trained. They're ejaculating preferentially and distributing more ejaculations with the scented female relative to the unscented. The non-receptive pair trained, right, where you got the odor meaning no, is actually now distributing ejaculations with the unscented female and giving her first ones. And the random paired are being random, as you would expect. Interestingly, if you notice the distribution of mounts and intermissions, they're also, in the non-receptive pair trained, distributing it with the unscented. It's as if the odor means no, and no is so bad that you're going to, even though she's receptive, you're going to go after the non-scented female. Okay, so they've learned both conditioned excitation and conditioned inhibition to the odor, which again also shows you the odor is neutral. So what is the effective UCS? What is the unconditioned stimulus? What is its nature? What do they have to do? I mentioned that ejaculation is important, and that's true for place preference, because with place preference, they experience the state of that in that novel place. So what did Stendhal say? If they remain in close facial contact, yeah, that's exactly what you see here. The PEI is a post-ejaculatory interval. That's their refractory period. If the male spends that refractory period with the female, so here he's ejaculating and we're pulling the female out. Okay, so he's not spending it with the female. But when he spends it with the female, you get the conditioning. And of course, with 2 ejaculations, that assumes that he's with the female after that first ejaculation. Okay? So yes, that's the period where learning happens, at least the learning that produces this kind of partner preference. What about females? We test both in my lab. So what about them? Turns out that the ability to pace the initiation or rate of copulation is necessary for inducing this orgasm-like response that then produces the pleasure of sex. We can do that 2 ways. We can do it in that bi-level chamber that you saw before, where she can control the male. Or we can do it in these uni-level pacing chambers, which are bisected. There's a male, there's a female. They're bisected by these little, by a plexiglass screen that has little holes. The female can get through because she's half the size of the male. He can't get through because the holes are too small. So for her, she can run back and forth from side to side in order to regulate her sexual contact with him. We can also do this with timed clitoral stimulation. So as I mentioned before, that induces both conditioned place preference and the partner preference I'll show you. The acquisition, like the males, is blocked by naloxone and not blocked by dopamine antagonists, which is suggestive of the fact that opioids, not dopamine, are the substrate of sexual reward. They're actually the substrate of all reward. Okay? Dopamine is just attention to reward-related cues. That's going to come important. So when we do the study, the same way we did it in the males, we have the paired group and a non-paired receptive, with the male being receptive or castrated. What we find is this. Solicitations, so here's the female making headwise orientation, and then she kicks him in the face and runs away. Here's the lordosis, this is how we score it, right? So you can see varying degrees of that. What you notice is first solicitation latency, lower in the paired group for the male that was scented relative to the unscented. First solicitation, more with the scented than the unscented, and solicitation frequency, way more with the scented than the unscented. So she's soliciting more, showing a partner preference. Not only that, who gets first ejaculation? The scented male. And she determines that, not him. So she's soliciting him, and then she's staying next to him. So he gets first ejaculation, and she shows more high magnitude lordoses when she's with her preferred partner relative to the other male. Okay, so she's not showing a Coolidge effect either. She's staying with the male that is associated with sexual pleasure. Okay? And that is kind of interesting. Not only that, but we can give them, what's cool about this paradigm is that we can give them a couple of reconditioning trials and then just put the odor on a gauze pad and drop that into their home cage. So, paired group versus unpaired, you can see when we look at neuronal Pfaus activation, that's these little black dots. This is a nuclear immediate early gene protein product that you find in neurons that have been activated. So we can simply look at the difference. Here's the piriform cortex, main olfactory cortex, medial preoptic area, ventral tegmental area. You notice this whole middle section of the VTA is absent in the unpaired relative to the paired. That's where all the dopamine neurons are that form the mesolimbic dopamine system. So they're absent. They're not activated in the unpaired, but they're activated selectively by the odor in the paired group. Okay, so this is, of course, what's mediating the behavior is the activation of these different systems. And you'll notice that there's more Pfaus protein induced in all of these different regions that are part of the bonding and reward circuit, right? And they're done more in the paired versus the unpaired group when we do the actual counts. Now what about clitoral stimulation? Yes, female mammals have a clitoris. And yes, it does the same thing in all female mammals. Okay? It is the same organ. It's shaped the same way. It's connected to a dorsal clitoral nerve that connects to the pudendal nerve that goes into the spinal cord and goes up to the brain and produces what? Pleasure. Okay? The glans penis is the same structure. Okay? And it also produces pleasure. So here's the clitoris, here's the vaginal opening, here's the anus, and here you can see that's a number 4 fine camel hair natural fiber brush. And it works, they love it. They do not like a synthetic fiber brush. It's prickly, they don't like prickly on their clitoris. I don't think we do either. Okay, so we have sexually naive females. They're given 3 trials of clitoral stimulation or control. In the control, we simply raise the tail, but we don't provide the clitoral stimulation. And we do the clitoral stimulation 2 different ways: masked, which is 1 every second for 5 minutes, or distributed, 1 every 5 seconds for 5 minutes, which is about the way that they would like to get their clitoris stimulated, between 5 and 10 to 15 seconds. Okay? So we do this, and what happens? This is what happens. When we do it masked, we get actual place preference. Here they're showing an increase for the light side and a decrease for the dark side, which they're not showing with the other stimulation parameters. Not only that, but when we look at different brain areas, we see that the CLS5, which is the distributed one, is activating the medial preoptic area. And it's activating the medial amygdala, the posterior dorsal ventral aspects, both the dorsal and the ventral. Here you can see the MEAPV, the ventral part, being activated by CLS1 as well, and with the control, no activation at all. Okay, so what does that mean? Well, here's the MPOA, here's the MEAPV. The MEAPV is a region involved in pleasure. So is the medial preoptic area. But remember that the 5 is different from the 1. This is going to prove to be interesting later on. We then did this. Now this is something you can't do in humans. When you want to know about humans' first sexual experiences, you really can't manipulate this. There's nobody that will let you do this. No mother and father will let you take their children and manipulate their first sexual experiences. So this is one of the things that we can do in rat models that we can't do in humans, and it's one of the reasons why this is so important, because we need retrospective, and people are notoriously bad at remembering things. What we did here was we dropped in a gauze pad with the almond odor, right, or water on it. And depending on the 2 conditions, we either had the gauze pad in when the sexually naive females received clitoral stimulation, or we had the water in when they received clitoral stimulation. In this case, the almond meant no clitoral stimulation was going to happen, so it would raise the tail, but they wouldn't get stimulated. And here, the water meant no. So, of course, we're doing the same thing, but instead of it being a non-receptive partner, we're making their first experiences with clitoral stimulation either be associated with the almond odor or associated with explicitly no clitoral stimulation with the almond odor. Everybody with me on that? And then we simply gave them their first sexual experience in the open field with 2 males, one that was scented, one that was unscented. All right? Here's what happens with the CLS unscented, this group. They solicit more the unscented male relative to the scented male. This is their first sexual experience. You might argue, well, wait, their clitoral stimulation was their first sexual experience. Yeah, I agree with that. I think masturbation typically precedes sexual interaction with a partner. That typically happens in our species. Probably it happens in their species as well. But here, you're seeing what the effect of that is on the expectation that they're giving in with their first experience. Here's the CLS scented, this group. And notice they're picking the scented male to solicit relative to the unscented male. They're doing exactly what copulation would have done, but now we're doing it with clitoral stimulation paired with the odor on a gauze pad. Yeah? Number of hops and darts, these would be proximal solicitations, follows the same rules. Who gets first solicitation also follows the same rules, and the male with the odor, because he's got an odor, and you can recognize that distally if you're a rat, they actually spend more time with him. What they don't do is pick that male for their first ejaculation. So they're showing a partner preference, but not a mate choice. So maybe the mate choice involves another sensory domain, and it does, called the cervix. And we'll get to cervical stimulation in a second. So this is the path of Cupid's arrow. I wrote it in Czech. I hope I wrote it correctly. But pleasure creates desire. Reward creates desire. When the food is good, you want more of it. When the food is bad, you don't. It's the same difference. Pleasure creates desire. So the question is, how? How does this work? And there are some rules that we've discovered. And the first one is this, that there's a primacy of first experiences with pleasure. The first time you tasted chocolate, it probably was a revelation. Why is that? Because it's the first time it happens. If you don't know what chocolate tastes like, nobody can describe it to you. They can tell you what the flavor is like, and you can maybe think about that, but you don't know what chocolate tastes like until you've tasted it. Likewise, you don't know what an orgasm is until you've had one, because your brain doesn't know that until it goes through the process of the sensory and motor activational pattern. Okay? So the primacy of first experiences is important. It's a very busy graph, but it's actually quite simple. We did what all Pavlovian people require you to do, things that are called sensory preconditioning. Right? We have a control condition. The animals get 10 trials to ejaculation, like you've seen before, and that's what you see here. So the ones that get scented females show a preference to ejaculate, and the number of ejaculations with the scented over the unscented. The ones who get the unscented female show a preference to ejaculate with the unscented female and distribute those ejaculations with her as well. What we do in the UCS pre-exposure is we give them 1 or 5 trials with an unscented female. So they copulate to ejaculation with a female that doesn't have the odor. Then we give them another 10 trials with one that does. Okay, so they've had now more trials with a scented female than with an unscented female. And then we simply put them in the open field and we test them again for the scented versus the unscented. So primacy of first experiences. What you see is that having one trial with the unscented completely eliminates the ability of the odor to acquire conditional properties. Okay? Why? Because that first ejaculation must be a huge thing for these animals. Probably like it is for us too. Okay? It's a big event. And that big event now ruins the ability of the odor to acquire conditional properties. Okay? Likewise, we can also give them CS pre-exposure. And what we do there is we drop in a gauze pad with the odor, the almond odor, once or 5 times. Yeah? And then we give them either training with scented or unscented, and we say, okay, who do you do? Well, when we do that, actually, okay. Yeah. The one trial with the scent does not have the capability of ruining the conditioning. Okay? The odor one time means nothing. But 5 times, and you have now produced what's called blocking. You've blocked the ability of the odor because the animals have learned that the odor means nothing. So you've blocked the ability, you've latently inhibited that ability of that odor to now acquire the conditional properties. Okay? Everybody see that? So the first experience matters. And first experiences with telling you that something doesn't associate with sex matters just as much as first experiences with telling you something does associate with sex. We've looked at it another way too, and this was quite by accident. In this particular study, okay, so here are the males that we would give to a female. Now notice the males are tethered. Why is that? Because when one is scented and the other's unscented, male rats are very cooperative when they have sex. So one will go, no, after you, no, please, after you, no, well, you just did, or please, it's your turn. And they crawl all over each other such that the scented male makes the unscented male now scented. Okay? If you've ever been in an elevator with somebody who wears way too much perfume or underarm deodorant or whatever it is, but eventually you spend the rest of the day smelling that person's perfume on your clothes. Okay? It's kind of the same thing. But what it meant when we didn't tether them is that both animals would end up being scented. So by tethering them, we can keep the scent to, the tether has a ring on the end of it and there's a spring, so he can only move this and he can only move that. So they're not going to scent each other, and the female can then choose to be with one or the other. Okay? Well, the student that was doing this, in Villa, had gone back to Mexico for Christmas, and somebody needed good studs, so I said, use Hanero studs. She did, and the studs wouldn't copulate. And I was like, that's crazy. Maybe the hormones are bad. We redid the hormones for the females. Tried it again, the studs wouldn't copulate. The females were perfectly in heat. So I was like, I don't understand this. So then we put them in the open field. They still wouldn't copulate. Okay? I'm on my bike riding home, and suddenly it hit me. The jacket. Oh my God, the jacket. So I turned my bike around, almost got hit by a car. The guy's screaming at me in French. I didn't even understand a word. I biked back, we put the jacket on, and they copulated perfectly well. Okay, that's crazy. So we did a study with this. So we asked, could a somatosensory cue on the individual control sexual arousal? In this study, we trained them either to have their first sexual experiences wearing the little rodent jacket or not wearing the rodent jacket. So that's the conditioning group, no jacket or jacket. And then on the final test, it was a 2x2 design. We simply had the jacket on or the jacket off, okay, in both groups. Let's start with the group that got no jacket. Well, having the jacket on or off didn't seem to matter for like condition level changes, which is kind of them saying, oh my God, I'm going to get laid, woo, in the 5-minute period before the female comes in, so they level change a lot. Didn't affect that. Didn't affect their intermission latency. Having the jacket on didn't affect it. Having the jacket off didn't affect it because the jacket means nothing to them. Didn't affect the number of intermissions or the number of ejaculations, okay? The group that had the jacket during their first experiences were affected. And you can see that here. Okay. Without the jacket on, level changes went down dramatically. Without the jacket on, the intermission latency went way up because they're not getting an erection. Number of intermissions went up, but that's because the number of ejaculations went down. These animals don't have arousal, or they have significantly less arousal without the jacket on. So we have serendipitously conditioned a little fetish in these rats. They need the jacket. They've made the association that the jacket is congruent with having sex, but with their first experiences, because clearly having the jacket on here doesn't matter. Okay? There's nothing about the jacket. There's nothing in their biological history, their evolutionary history, that has anything to do with wearing a jacket. Okay? And they're not weird. What's weird is that you can get this association at all in an animal that kind of shouldn't be showing it, but does. Okay. So then we said, is there something about the jacket, something about yes meaning yes and it being really big? Can we make the jacket excitatory or inhibitory? And the way we did this here, we kind of killed two birds with one stone. With the jacket on, they got receptive females, and we did the sequential conditioning. Jacket off, they got non-receptive females. So here the jacket is clearly now associated with yes. Here, with the jacket on, they got non-receptive females, and jacket off, they got receptive females. So the jacket is associated with no. So they know when they're wearing a jacket, it means that the females are going to beat the crap out of them if they try to have sex with them, so it's not going to work. We then also varied the first experience with the jacket on or off in these particular conditions, right? So you got 4 different first experiences. Jacket on means yes, jacket on means no. Jacket off means yes, jacket off means no. Okay? We finally do a final test with the jacket on. Everybody with me on that? So let's first look at the excitatory group. So here's the excitation, no jacket start, jacket start. Right? So remember, no jacket start in this group meant they got a non-receptive female. Jacket start meant they got a receptive female. But there's no difference for number of intermissions, but there is a difference for ejaculation latency and number of ejaculations. And it's in the right direction. That is, when they got no jacket start, the ejaculation latency goes up. Because they've made an association that, well, okay, if I wear the jacket means yes. No jacket means no, but that was their first experience, right? So there's a little bit of a delay. Not only that, but they show fewer ejaculations relative to the ones where the jacket meant yes and that was their first experience, right? And the post-ejaculatory interval went down in that group as well, okay? Which the post-ejaculatory refractory period is usually the hardest, most set in stone sexual behavior that you can get. You almost can't do anything pharmacologically to manipulate it. I wish you could, because that would be a drug that would be amazing, but you can't. But this does for some reason, okay, which is interesting. Let's look at the inhibition group. Yeah, jackets start, number of intermissions goes up. Why? Because the number of ejaculations goes down, because the ejaculation latency goes way up. So when the jacket was the first thing, and it meant no, even though no jacket was the second thing, which meant yes, they clearly were affected by that first experience. Okay? Such that now the jacket means no, and no is a serious thing. Right? Rats know what no means, and they don't persist. And something like a jacket can actually now reinforce that even better. Okay. The jacket can also be a discrete cue on the female. So we asked the question, okay, why use odors when you use a somatosensory cue? So here's female with the jacket on, female without the jacket, there's the male. So what happens? Choice for first ejaculation. The paired group, when they received their first and subsequent 9 sexual experiences with a jacketed female, sure enough, they're showing a preference to ejaculate with the jacketed female. Right? And of course, the unpaired group aren't. But they're not showing a preference for the unjacketed. Right? Why is that? Because the jacket is a distal cue that they can't see very well, and probably doesn't have an odor on it. Right? So in subsequent work, we want to see whether the odor, if the odor means yes and the jacket means no, which cue, one that's distal or one that's more proximal, will take over. Right? So essentially, when it's a contextual cue on the individual, it affects sexual arousal. When it's a discrete partner cue, it affects partner preference and not arousal per se. Okay? So when it's on you, that's a contextual thing. When it's on the partner, that's a discrete partner cue. It's different. All right. Rats will also sign track. This is something that's big in the drug addiction literature. When you have a cue that indicates the availability of a drug, what do animals do? Well, animals break into like little thirds. Some of them become real cue conditioners or sign trackers, where they just go after the cue, and after the cue, they stay with the cue. They want to hang out with the cue. They'll do anything for the cue. And then when they go get the drug, they take way more drug than animals that are goal trackers, who once you show them that you cue them in, they go over to the goal and try to get the drug. They take way less amount of drug than animals that are sign trackers. What about sign tracking for sex? Well, we use these little football cones. Okay, we had a CS zone and then a US zone, a UCS zone, where the animal would copulate with the female. Okay, so we put this in for a couple of minutes, and then we present the female. So this becomes now a cue that tells the animal the female will be here in 2 minutes. What do they do over trials? Well, when the cue is in the CS zone, okay, versus when it's not. When it's not in the CS zone, they stay here waiting for the female. You can see that over the course of 12 sessions. However, when the cue is there, okay, here's first session, 6th session, the US zone and CS zone. But by the 12th session, they're simply hanging out with the cue, all of them. Not some of them, all of them. And even though the female is present, they're mounting the cone, which is insane. Okay? It's insane. They could just run over here. This is not a big open field. They could just run over here and go through a hole and get to the female. But they're not. They're hanging out with the cue. Okay, Earth boys are easy and dumb at the same time. This is nuts. This is crazy behavior. But it's not so crazy, and you'll see why in a little minute. Okay, rule number 2. Opioids are the substrate of reward and the substrate of pleasure. How do we know? Well, I kind of gave you the hint of that before, but we can block the rewarding effects of sexual stimulation in both males and females with either an opioid antagonist, like naloxone or naltrexone, or frustration induced by the presentation of a sexually non-receptive partner, or one that's inaccessible. All 3 of those things block reward. So they're like non-reward paradigms that can be used. I'm only going to talk about naloxone because it's easy to identify the opioid action of that. So here's what happens in males when we either give them their first sexual experiences under the influence of saline or under the influence of naloxone. Okay, these animals are tested every 4 days. That's the female cycle. So we do this for 9 trials, and then on the 10th trial, we give everybody saline. Okay, so it's an expectancy. If the blockade of opioid receptors produced a state of non-reward, then they're getting a saline injection. Then that state, that expectancy of non-reward, should now be brought out, even though there's no drug on board. And that's exactly what happens. Look at the saline group. They showed the ejaculatory preference for the familiar over the novel female. That preference is gone with the animals that previously had been trained under naloxone. Yeah? If anything, they're now showing a preference for the novel. So now they've got a Coolidge effect, because the other one didn't work. Or at least they ejaculated, but the ejaculation didn't produce the pleasure that would be involved in, say, an orgasm. Okay? Same thing for the jacket. Here's the animals trained with saline. They're showing the effect for the jacketed female over the unjacketed female. Here's the naloxone group. They're showing now a preference for the unjacketed over the jacketed. Right? So for the male, at least, it's shifting the preference to the other female. All right? It's worse in females. But before we get to that, let's get to what opioids are doing and where. Right? So mu opioid receptors exist in all regions of the sexual arousal, reward, and bonding circuit in the brain. Opioid actions sensitize dopamine neurons and cause greater dopamine release in the presence of cues that are associated with reward. And opioids do this through a very well-characterized molecular mechanism. We know that whole brain beta endorphin content increases after ejaculations in male rats. And we know that in the medial preoptic area, opioids inhibit copulation. Stress-induced opioids can actually be inhibited by naloxone to facilitate copulation if the opioid releases by stress. And we know in the MPOA that mu opioid receptors internalize, something that they do when they're bound. They are brought in under the membrane so that the opioid can be broken down and the receptor can be recirculated. Whereas in the ventral tegmental area, the area that I told you the dopamine neurons exist, opioids sensitize dopamine release. Okay, they actually have a calcium binding molecular mechanism that makes more dopamine be released in the presence of cues that are associated with opioid reward. Okay? And we know that naloxone here can inhibit conditioned appetitive behavior. So we asked the question then, what happens when we put naloxone directly into those brain areas? So we inserted cannula into the POA, gave them either saline or naloxone, as we'd done systemically. And what happened? Exactly the same thing we saw systemically. With saline, they show a preference for the familiar over the novel. With naloxone, they show the preference for the novel over the familiar. Okay, so the MPOA, naloxone, is doing exactly what it did systemically. Whereas, and then it did the same thing for the jacket. Right? So we see the preference for the jacket over the no jacket in the saline infusions, preference for no jacket over jacket with naloxone infusions. Yeah? When we looked at the ventral tegmental area, it did something a little bit different. So if the MPOA is valencing the reward cue, the MPOA is saying salient or not. Not reward or not, but salient or not. Yeah? Because we simply, with naloxone, knocked out any preference. We didn't shift the preference. We just knocked it out. It's not salient. Because dopamine is telling you about the salience of a cue that's related to reward. It's not telling you about the reward. Whereas the MPOA is telling you about the reward, and it's relaying that information to dopamine neurons through projections from the MPOA to the VTA, right? So it did it for the odor. It did it for the jacket as well. So identical findings. In addition, Larry Young's group found that mu opioid antagonism disrupted sociosexual partner preference in male prairie voles. Right? We think it's all about oxytocin and vasopressin. It is, but it's also about opioids and reward. So what you see here is the effect of naltrexone, right, on partner preference. Right? 3 pairings, and it's gone. Right? And of course, the prairie vole, as you probably all know, is like the animal model of monogamy and love and oxytocin-induced partner preference and all this kind of stuff. But you can knock it out just like you can in rats with naltrexone, a longer-acting opioid antagonist. Not only that, you can knock it out with a selective mu receptor antagonist, CTAP. Right? But you see, knocked out here, right, when it was put directly into the nucleus accumbens, right? Here's the CTAP effect. But here's the CTAP effect in the caudate putamen. So just a little bit, a couple millimeters north, and you get a complete wipeout of that effect. Okay, so the male prairie vole depends on sexual reward, just like the rat does, even though theirs is allegedly not conditional. Okay, what about females? Well, we do the same study in females. We give them their first 9 conditioning trials with saline or naloxone, followed by an open field test, and on that day for the open field, everybody gets saline. So again, it's an expectancy of non-reward versus reward. And what happens on that final test? Well, here's look at solicitations, pacing-related male, no pacing-related male. You see the beautiful effect with saline. It's gone with naloxone. Also, the females receive fewer intromissions and fewer ejaculations. And why is that? Well, it turns out because they're not lordosing a lot, and they're fighting. Here's the different lordosis magnitudes, and you can see that with naloxone, relative to saline, you've knocked out the high lordosis. And that's critical, because in a male mouse, in order to get vaginal penetration, she has to show a full lordosis. When she doesn't, he can't get in. So, consequently, they're getting fewer ejaculations. And it turns out that if we actually just look at all the behaviors that the females show under these circumstances, here's the solicitations for saline versus naloxone, hops and darts. She shows way more defensive responses in fighting. And that, mixed with the lack of solicitations and the lack of high lordosis, means she's getting fewer intermissions and fewer ejaculations. Now, why is this so interesting? Because she's fully primed with estrogen and progesterone. She should be out of her mind. But her experience, not the hormones, her experience is now overriding the hormone effects. And why is that so interesting? Because clinically, I mean, I ran into this meme, and clinicians support this idea. It's funny how men say that even bad sex was good at the time. Not so for women. Bad sex was really bad at the time. Okay, it's not like you just say, well, I'll do another part. No, sex sucks. You don't like it anymore. Because it feels bad or it doesn't feel good. And that has a profound effect, much more so in females than in males. Because males will simply go to the other one. Whereas the females aren't going to the other one. They just don't want to have sex anymore. Okay? And from a clinical standpoint, that's very important when you think of a desire disorder as being the #1 thing that women complain about. And why do they complain about that? Because they're frigid? No, because they have bad sex. And they learn. Their brains accommodate to that. Okay? Rule number 3, pleasure activates oxytocin and vasopressin, which act as substrates for social bonding and arousal. Okay, so I told you about the opioid effect. That's necessary for the UCS. But now you're talking about what opioids are doing. Okay, so we did a study where we looked at the almond odor, CS for CEP, and how it might activate oxytocin and vasopressin neurons. We did the same kind of conditioning, bi-level chambers that we did. We did the open field test. We did 2 reconditioning trials. And then, as I told you before, the power of this paradigm is that you can present the odor alone. We presented the odor alone, gave them exposure for 1.5 hours. We perfused, did immunohistochemistry, double labeling for FOS within oxytocin neurons or FOS within vasopressin neurons. Okay? And we looked at the populations in the paraventricular nucleus of the hypothalamus and the supraoptic, the places where those neurons are. And sure enough, when we find FOS within oxytocin cells, there's a significant increase when the odor was paired relative to the odor unpaired in the paraventricular nucleus, this region here. And for the supraoptic, which you see here, right, here's the difference between paired and unpaired, supraoptic for vasopressin, but not oxytocin. It's a bit of a double dissociation. Yeah? So what does that mean? Well, it means that oxytocin here, which is largely projecting to the spinal cord and to the rest of the brain, okay, these ones also project to the spinal cord, but they also project to the posterior pituitary, are differentially activated. When the odor is paired, you're activating the oxytocin neurons that are going to be projecting to sites like the ventral tegmental area, the nucleus accumbens, that are important for bonding. Okay? So if the male can recognize the female, that means he's bonded. And oxytocin is clearly playing a role in that. We also did this study. So we know that one conditioning trial doesn't, even though it has a profound effect on, if they're with an unscented female, on the ability of the scent to activate it, we also know that they don't show a conditioned ejaculatory preference after only one trial, right? But here we ask the question, if we gave subcutaneous oxytocin, vasopressin, or saline, would that, before that first conditioning trial, would that actually potentiate? So in other words, could we pharmacologically move the animals to as if they've had 9 conditioning trials by having oxytocin or vasopressin on the first trial with saline as the control? Everybody with me? Good? Visual feedback? Nod your head? Yeah. Okay. So we do that, give them the first trial, and 4 days later, we give them the sexual preference test with no injection. So here what we're asking is, pharmacologically, did we do what the conditioning did to those oxytocin neurons? Activate more oxytocin release? Would the conditioning kind of circumvent that? Or would the oxytocin here, or vasopressin, circumvent that? Yeah? And here's what we found. Sure enough, saline group had no preference, which we expected because it was only the first trial. Vasopressin group didn't either, but the oxytocin group, Gesundheit, showed a significant conditioned ejaculatory preference. Now, I'm not suggesting anybody should go out and buy oxytocin nasal spray on a first date, but that's kind of what this is suggesting, right? That you activate this conditionally with experience, but you can get a drug effect to produce the same kind of priming. Okay? That's really all drugs do. They prime the brain to be responsive to things, and that's what oxytocin peripherally is doing here. We also did, so then we said, okay, look, let's go whole hog. Let's do some autoradiography. What are oxytocin receptor populations? Are they affected by conditioning? It turns out they're not. Well, I shouldn't say they're not. A male rat's first ejaculation, these rats either had one ejaculation paired with the odor, ejaculation unpaired, or we contrasted this with sexually naive animals, right? And 3 regions that we found for oxytocin receptor that were activated regardless of the conditioning group, but by ejaculation. So the MPOA, the medial preoptic area, was activated in both the paired and the unpaired relative to the naive. It was significantly greater. Likewise, in the ventromedial hypothalamus, it was activated relative to the naives. And the central nucleus of the amygdala was activated relative to the naives. So it's kind of like a 1-2 punch with oxytocin. First ejaculation is, through an epigenetic mechanism, activating oxytocin receptor. Subsequent ejaculations that are associated with the odor now allow the odor to activate oxytocin neurons, which are going to be projecting to these oxytocin receptors and unloading oxytocin on them, right? Along with other receptor populations, but we didn't find an effective ejaculation on. Okay? So this kind of hones in the areas that we need to look at for oxytocin's effect. And then this happened. Amanda Hawley in my lab, former grad student, found that female rats actually show conditioned mate guarding. Now, mate guarding is a gold standard of monogamy. Prairie vole females show it, who are monogamous. The promiscuous females, like montane voles, don't show it, and it's all related to oxytocin. Well, in this particular cast of characters, we either have a normal female-female competition where the females basically run astride one another and they compete each other out and they kind of intercept the male. More dominant females tend to intercept the male when he's giving off his I'm going to ejaculate call, which is 50 kilohertz call. Whereas in this one, you have a girlfriend-dominant competition, right? So you have 2 females, but one is the one that's paired with the male, who now has a monogamous bond with that male, and the other one would be a competitor female. So we just did the same thing. We had the males and the females, where the females paired with the males. Okay, the males have an almond odor. We have them paired with the males. So he's a boyfriend, she's a girlfriend, and then you got a competitor drop in. So what does she do? And it's insane. Here's what she does. She, the girlfriend, will hover and present to the boyfriend. She hangs out with him. The competitor typically stays away, but sometimes she tries. And when she tries, she'll come close, because she's going to solicit. When she does that, the girlfriend actually gets in between them. So it calls behavior blocking. Okay, she blocks the other one. And if the other one is, because they're both sexually receptive, so if the other one kind of comes around and does a solicitation, before the male can go, what? The girlfriend is mounting the competitor. And that's not a copulatory mount. That is an aggressive dominance mount. Okay? There's no pelvic thrusting. If it was a copulatory mount trying to show the male what to do, she'd pelvic thrust. But here she doesn't pelvic thrust. She actually like puts the female's head into the wall. Okay? And after she does this enough times, if you're using the same competitor with the same girlfriend, guess what? The competitor doesn't go anywhere near them. So again, despite the fact that she is in heat and should be showing all those competitive behaviors, she learns that she's going to get beat up by the girlfriend. So she doesn't go near the male. Okay? If it wasn't the girlfriend's male, it was a different male, guess what they would do? They would, oops, hang on. They would show these patterns. Okay? So it's not that they're monogamous or promiscuous. When she's with the boyfriend, it's a different context than when she's with another male that she can play with. Okay? And likewise for the competitor. So Amanda looked at phospholipid and oxytocin neurons when she presented the odor that was associated with that. And sure enough, in the paired group, you get increased phospholipid and oxytocin neurons in both the SON and PVN. Bit of a sex difference there because we didn't see it in the SON in males. And phospholipid and vasopressin in the SON, but not the PVN, so that's not a sex difference. Okay? And then she did what we had done before, gave oxytocin, vasopressin, or saline on the first trial, and then tested them 4 days later, okay? As we had done in the males. So here you can see mate guarding after saline, paired or unpaired, all these different behaviors. Right, here's the blocking behavior. Here's the hovering and presenting behavior. No difference, no effect. Because again, one conditioning trial is not sufficient. But what you notice with oxytocin, she hovers and presents significantly more when it was paired with the scented male. Now with the odor, she gets more intromissions, and she's also soliciting more. And that makes sense because that's what oxytocin does. It removes that social distance between the animals. Right? With vasopressin, on the other hand, you didn't get the effect on hovering and presenting, on intromissions, or on solicitations, but you got the effect on the competitive blocking. So when vasopressin is acting as a vasopressor and is increasing your awareness of the competitor, you get an increase in competitive blocking. Okay. We also find that optimal pacing conditions in those unilevel chambers activate oxytocin neurons. Right? So when it's one hole, the females actually learn something, and they show greater oxytocin, or greater FOS within oxytocin neurons in the paired versus the unpaired group, in the PVN, in the SON, not in the VTA, in the medial preoptic area, and in the ventromedial nucleus of the hypothalamus. Okay. And this is a real clincher. In males, remember I showed you that sign tracking data before, yeah? When we train them with saline or oxytocin, we turn the sign trackers into goal trackers. It is as if oxytocin has changed the nature of the reward, has changed the way the brain interprets the goal relative to the sign. And could this be used for drug addiction? I don't know, maybe. And might turn the sign trackers, who take way more drug because they're way more aroused to the cue, into goal trackers. But oxytocin does this, and that's an amazing finding. Yeah? It seems to be personifying the reward, taking it away from the object and turning it into a partner, and making the animal go after the partner. So that's kind of cool. Rule number 4, stimuli associated with pleasure activate mesolimbic and hypothalamic dopamine, and that in turn, those mechanisms act as substrates for attention, social recognition, and desire. So, same kind of study that we did before, paired males versus unpaired, do the sexual preference test, do 2 reconditioning trials, and then, instead of just dropping in the conditioned odor, we drop in the conditioned odor, we drop in a novel odor, and we drop in estrus female odors. How do we get those? Really simple, a wet Q-tip, vaginal secretions from a sexually receptive female placed on a slide. Okay? Male rats love those. It's okay. Male rats love those. So we expose for 1.5 hours, and then we collect in that 1.5 hours microdialysis samples that are collected every 15 minutes. Right? So we're collecting from the nucleus accumbens here, and we're asking the question, does dopamine go up? Yeah. Well, in the paired group, the almond odor, which was paired, yep, it produces an effect that's like about 10 milligrams per kilogram of cocaine. I'd say love is the drug, but that's kind of annoying. And it does it in the paired group, but not in the unpaired group. Yeah. The receptive female odor does it in both. Not to the same extent, perhaps, but it does it in both, as it should, because it's a salient cue. And the lemon odor does nothing in either group. So it's not just that the males have a kind of a thing for odor. They have a thing for a particular odor that was associated with sexual pleasure. It turns out as well, dopamine can facilitate familiarity in male prairie voles. So they typically do the study where the male is either with the female for 24 hours with mating or 6 hours of cohabitation without any mating. We'll notice that the mating produces the partner preference, the cohabitation does not. However, if the cohabitation is under the influence of apomorphine, which is a dopamine agonist, you get the partner preference. And in fact, haloperidol, a dopamine antagonist, can destroy the partner preference that's produced by the 24 hours with mating. Okay? And for the sake of time, I won't go into that. But it turns out it's a D2, well, maybe I will. It turns out it's a D2-like effect. What happens here? Brendan Aragona gave a D1 versus D2 agonist and found that, yeah, the D2 agonist produced the effect the D1 agonist did not. So it's a D2 effect. We know that dopamine type 2 receptors are associated with certain second messengers. And here, which is in the striatum, they're associated with protein kinase A. Okay, through a G protein coupled mechanism, you activate protein kinase A or you deactivate it. Well, D2 binding deactivates it. And when you deactivate it, right, with a selective drug that deactivates protein kinase A, you get the effect. When you activate it, you inhibit the effect. The D1 activates it. So Brandon has made a suggestion that D2s facilitate pair bonding by kind of opening the system back up, and that when the system closes after reward, it's now become a D1 effect. And sure enough, when you look at animals that are already pair bonded, they have increased D1 binding and decreased D2, and vice versa for virgins, who have more D2 than D1. Okay? So that's kind of cool. Turns out that quinpirole, which is a D2 dopamine type 2 agonist, if we do the same thing here, but we use rats, and we pair a male with a male, or a male with a female, under the influence of saline or quinpirole. Now I'm just going to show you the male data, but here is the total time spent in contact with a female under saline versus the male. With quinpirole, they spend more time in contact with the male relative to the female. Okay, so they're with the male for 6 hours under the influence of quinpirole, and on the next test, we give them the choice between a male and an estrus female, right? They should go to the estrus female, but no, they go to the male. And when they go to the male, what do? Well, when they're with the female, under the saline circumstances, they're showing non-contact erections in her presence. That's perfectly normal behavior. However, when we've done it with the male, now they're showing non-contact erections in response to the male. Is it a measure of fluidity? Is it a mechanism of fluidity? I don't know. But we're definitely going to follow up on that. Rule number 5, and this is the last rule, inhibition of demethylation. Okay, here we get to the Cupid's epigenetics. Acts like mu-opioid receptor antagonism. That is, we block the epigenetic mechanisms that reward induces, acts like blockade of reward. How do we do that? We did it with a drug called Oryzon, which is a lysine-specific dimethylase 1A antagonist. Okay? And why that? Because that's a methylation phenomenon that exists within oxytocin and dopamine neurons. So we were interested in that. Luckily, that exists. It's a flavin-dependent monoamine oxidase, which then demethylates mono and dimethylated lysines. When we do that, and we do that during conditioning, so it's like we did with naloxone, now we're doing it with Oryzon, what happens? This is what happens. When we look at the initiation of that hovering and presenting behavior, the animals that got saline paired are showing an increase relative to saline unpaired. That makes sense. They're also showing more blocking behaviors. But with Oryzon, we've knocked it out completely, as we would have with naloxone. So non-reward, or not being able to activate the mechanisms that reward is activating, does the same thing. Okay. Not only that, but as we expected, it knocks out the Pfaus within oxytocin neurons and within vasopressin neurons. Okay, so here's the paired saline, LSD1 paired, unpaired. So it produces an effect as if the pairing never happened. Okay, so I asked you how, I showed you how. But you could still say, but how? Why would any of this work? And these are rats, for Christ's sake. Why does this work for rats? The first reason is this, right? CD38 gene, which is a cluster differentiation at 38 kilodaltons, is located on chromosome 4. It's a very important gene because it codes for a protein, an NAD converting enzyme, which is a transmembrane glycoprotein that I'm sure you all know, opens up ryanodine-sensitive intracellular calcium channels. So it allows calcium to kind of come out of the endoplasmic reticulum and into the neuron itself. Why is that important? Because calcium binding proteins are critical for the release of neurotransmitter. Yeah? And they're driven by calcium. Well, within oxytocin neurons, that protein is absolutely critical for oxytocin release. We know that in CD38 deficient mice, the pathway isn't working. Oxytocin secretion is whoppingly impaired. Those mice show deficits in maternal, social, and sexual behaviors. Okay? So that's one thing that happens. But it has a relationship to mu-opioid receptor activation and internalization. Mu-opioid receptors, when they're activated, they activate a GI protein that wipes out cyclic AMP and activates MAP kinase signaling. So, why is that important? Because activation of ERK in particular increases the transcription of CD38. And through this kind of MEK2A and ERK vectors causes mRNA stabilization so that you can actually then translate into a huge amount of protein. So mu-opioid receptors within oxytocin neurons are activating transcription and translation of CD38 protein. That's critical. That's absolutely critical. In addition, opioids, like I mentioned, sensitize mesolimbic dopamine transmission. Now, in the short run, they inhibit it. All right? And part of what happens when a male rat ejaculates is you get an immediate decrease in dopamine release. You can see that here. These are the points where this male, this is voltammetric data, but here you see where the points mid the post-ejaculatory intervals. Dopamine goes down, and then it goes back up again. So it's, as you break down and internalize the opioid receptor, you get rid of it. Now the inhibition on dopamine is released, and so dopamine goes back up. Here's the point of ejaculation, it's gone down, the midpoints. So we know that happens. The hypothalamus, the arcuate nucleus, sends endorphin neurons to the ventral tegmental area, where they do two things. They inhibit these GABA interneurons that normally keep dopamine in check, and they inhibit dopamine neurons. But by inhibiting these, ultimately, you build up a sensitized dopamine response. Okay? And that's also critical. So when you put it all together, you have increased attention toward incentive stimuli that are associated with reward. And in this case, it's partner-related cues. So let's put it all together. What's oxytocin and dopamine doing? Well, dopamine is activating oxytocin. There are dopamine projections onto oxytocin neurons in the paraventricular nucleus. And sensitized dopamine then would be carrying the conditioned stimulus Q onto those oxytocin neurons, where it's doing what? Opening up a calcium channel. So it's depolarizing the neuron, calcium's rushing in, calcium binding proteins are going to be driven by calcium to now cause oxytocin release. And that's all well and good. However, opioids also bind to those oxytocin neurons, where they activate CD38, activate calcium binding proteins. The two things together form a kind of molecular Hebbian synapse. They're firing together, and when they do, now you're recruiting more and more oxytocin neurons, which are now releasing more and more oxytocin within limbic structures, the hippocampus, and the spinal cord. Right? So here you see it's going to ventral tegmental area, it's going to the hippocampus, it's going to the spinal cord. What is it doing? Well, it's preparing the body to interact with the cues, because it's driving sexual arousal and blood flow to the genitals. Oxytocin has parvocellular and magnocellular divisions, okay, that go to different places. But conditioned oxytocin activation links conditioned arousal to the CEP. And it does so by activating in the spinal cord parasympathetic now activation that causes genital blood flow. And it does that in males and in females, causing clitoral blood flow in females, or vaginal to clitoral blood flow. Turns out as well that PVN oxytocin neurons project to the lumbar spinal cord and activate neurons that induce that blood flow, but they also, potentially from the release from the pituitary right before orgasm, activate these SAN, SNB neurons, which are part of the spinal ejaculation and orgasm generator. This is the generator that converts the parasympathetic to sympathetic. Okay? And it kind of, as the sympathetic arousal is increasing, right, you get closer to orgasm. When orgasm happens, as a spinal reflex, you now convert and switch, which is what these neurons do, convert and switch from parasympathetic to sympathetic, right? And now blood flows back into the core. You crimp the blood vessels, or you open them up and then you crimp them, so blood now stays in the core and you lose your erected clitoris or your erected penis. So the question is, are those contributing to this? And it turns out that indeed they are. One of the things that we've recently found, so here's a male rat, here's a rat, here are the kind of the way the nerves are getting into her brain, but she's receiving from his penile intermissions two types of stimulation, clitoral stimulation and vaginal cervical stimulation. And it turns out that oxytocin receptors exist on every sensory neuron in the clitoris. So what's oxytocin doing? It's helping to open the clitoris up during erection, opening up those thousands of nerve endings, which are now open to tactile stimulation. It does that as well on the cervix. Oxytocin, here's the cervix. Oxytocin receptors exist on the cervix. And what does oxytocin do to the cervix? What does it do to the uterus? Anybody know? Oxytocin. Okay, you're dilated at 12 centimeters. Baby's trying to get through, can't because you need to be dilated a little bit more. They give you what? Pitocin, which is oxytocin. And what does that do? It causes uterine contractions and further cervical dilation. Right? It causes essentially what's called a Shafiq reflex, which is an upsuck mechanism. Okay, so where the cervix actually kind of dials down into the sperm pool and sucks it up. And the uterine contractions allow the sperm tails are good, but they're not that good. Uterine contractions help the sperm get up the cristae of the uterus into the fallopian tubes where they can get to the egg. So oxytocin receptors in the cervix would be activating that mechanism. And oxytocin peripherally delivered to the cervix activates that mechanism. So think about this for a minute. Conditioned release of oxytocin to the periphery in the initial phase of sex is going to drive faster clitoral arousal. It's going to make the Shafiq reflex more likely to occur when female rats have orgasms. And that is going to be displayed then, and we've discovered that it is displayed as a faster termination of estrus when the females are with their preferred partners as opposed to their non-preferred partners, or a partner that they don't even know. Okay? So there's a faster decrease of solicitations after the vagocervical stimulation. And that's enhanced by peripheral oxytocin, which thus ensures paternity. Okay? So, how do we know? Because paced copulation is what they find rewarding, and sure enough, if they have paced copulation, you get 5 intermissions or 10 intermissions, paced versus non-paced, they show more of them get pregnant in the pacing condition relative to the non-pacing condition. And as if that wasn't good enough, it turns out that when we do clitoral stimulation with a vibrating Q-tip, right? I'm almost done. When we do clitoral stimulation with a vibrating Q-tip, yeah, we either do it masked, we do it distributed, or they get none. What happens? It's really simple. They show increased solicitations to the one that they like. They show less solicitations to the ones they don't like, but here's the masked group, here's the distributed group. If they get more than 9 intermissions that has been preceded by this 1 every 5 minute vibrating stimulation of the clitoris, more of them get pregnant than the others. All right? So this is a really important finding because taken together, we now understand something. Pleasure activates bonding, attention, and arousal to create motivation, desire, and attraction. And it does so at the basic substrate of opioids from the arcuate, activating these different regions, activating oxytocin, dopamine, and vasopressin in different ways. Okay? Sensitizing dopamine, but activating oxytocin, vasopressin, and activating calcium binding proteins there that help not only to increase arousal, but help to increase bonding and attention to those cues. Yeah? Such that when you become aware at the cognitive and emotional level, in the cortex, presumably, you're aware of your attraction and arousal and desire. And you know which ones are hot and not. So, in summary, why do we have sex? Is it to make babies? Yeah, ultimately, but proximately, no. It's for fun. It's for pleasure. And perhaps all species on this planet have sex for pleasure most of the time, which is proximate, not ultimate causality. Mating can occur or not with a bit of luck in a mating context, but mating is not critical for the individual. No one dies from not having sex. In fact, sometimes you die because you have sex. So long as enough individuals in each generation of a species are successful at it. Sex doesn't have to serve reproduction. It does. It's kind of icing on the cake, but you have the cake, and the cake is fun, is pleasure. So desire for whom an individual mates with can work hand in hand with the desire for sex, such sexual bonding, even for a short time, right, where you have short pair bonds, requires conditioning and expectation of sexual reward and pleasure to produce effective appetitive responses toward the preferred partners. And first experiences with sexual reward matter. And this kind of captures everything I've just been telling you, right? So it's insane. And with that, I'll say dikwi and thank my former grad students and current grad students in the granting agencies in Canada and Mexico that supported this research. I still have 2 more to graduate from Concordia, but the rest have gone on. And these are actually pictures of them now, as opposed to everybody always has pictures of them then. But these are pictures of them now, okay? And I'm very, very proud of them and their success. And I'll thank you and take any questions you might have.